In this screencast, our goal is to review the three types of neurons and their general structure and basic functions. Neurons are like other cells in many ways. They have a nucleus with DNA, mitochondria, endoplasmic reticulum and ribosomes, Golgi apparatus, etc. They differ in that they're specialized for communication and transmission of information. The nervous system performs three basic functions. It takes in sensory input via sensory receptors and sends this information via an afferent pathway to the central nervous system. There, this information gets integrated or interpreted, and a decision is made about what to do. And finally, a motor output is sent to the effector organ, in this particular case a skeletal muscle, via an efferent pathway to execute an appropriate response. To perform these functions, the nervous system has three basic types of nerve cells, or neurons. Sensory neurons, or afferents, that transmit information from the peripheral sensory receptors to the central nervous system. Motor neurons, or efferents, that transmit information from the central nervous system to the effector in the periphery. And interneurons, which are neurons that reside completely within the central nervous system. A given neural circuit will have one sensory neuron at the beginning and end with a motor neuron, and may have anywhere from 0 to many interneurons in between. The general structure of a typical neuron, including sensory neurons, interneurons, and motor neurons, consists of the dendrites, which is where information comes into the cell, a cell body or soma that contains the nucleus, the spike initiating zone, also known as the trigger zone, where action potentials are initiated, an axon, down which action potentials propagate, often long distances, to other cells, and this axon may be myelinated or wrapped in myelin, with gaps between the myelin known as nodes of Ranvier. Finally, neurons have a presynaptic terminal, also known as the axon terminal, which is the location where a neuron passes along information to the next cell, either a neuron or an effector cell. A note about the spike initiating zone. Sometimes it's also referred to as the axon hillock, as is shown in this figure. However, an axon hillock is an anatomical term, not a functional one, and not all neurons have an axon hillock. Thus, in this class, we will preferentially use the term spike initiating zone or trigger zone. Because neurons have different functions, they have different shapes or morphologies. Some may have just a few dendrites or a very simple dendritic tree, while others that receive lots of inputs may have a lot of dendrites or very extensive dendritic trees. For example, Purkinje cells of the cerebellum receive about 250,000 inputs from other cells and have lots and lots of dendrites to receive all these inputs. Even though neurons can vary extensively in their morphologies, most have 4 basic functional regions in common, and each region produces a characteristic signal. The first functional region is the input region, which corresponds to the dendrites, where information comes into the cell and graded potentials are produced. The second region is the integrative region, which corresponds to the spike initiating zone, where graded potentials can trigger the initiation of action potentials. The third region is the conductive region, which corresponds to the axon, where the action potential propagates along to the last region, which is the output region, which corresponds to the presynaptic or axon terminal, where the cell produces an output signal, typically the release of neurotransmitter. Now let's look at each of these cell types a little more, starting with sensory neurons. All sensory pathways have certain elements in common. They begin with a stimulus, such as light, sound, odor, pressure, temperature, etc., that acts on a sensory receptor. Each sensory receptor is sensitive to a particular type of stimulus. The receptor is a transducer that converts the stimulus into an intracellular signal, namely a change in membrane potential called a receptor potential, a type of graded potential. We can further differentiate sensory receptors into the central receptors that are in or lie close to the brain, and these are the receptors that mediate the 5 special senses: vision, hearing, equilibrium, smell, and taste. The second basic type of sensory receptor are the peripheral receptors that lie outside the brain. And these can be chemoreceptors that detect pH and changes in chemicals and gases, osmoreceptors that respond to osmolarity, thermoreceptors that respond to temperature, baroreceptors that respond to pressure, proprioceptors that respond to body position, and other mechanoreceptors such as those that respond to pain, vibration, and touch. Most of the sensory receptors for the special senses, with the exception of smell or olfaction, are unusual in that they involve a non-neural cell that transduces the stimulus into a signal and the release of neurotransmitter onto a sensory neuron. However, for the most part in lecture, we'll focus on the somatic sensory systems that start with a sensory neuron. And the four somatic senses, or somatosensory senses, are touch, proprioception, temperature, and nociception, or pain. For proprioception, we'll focus on the muscle spindle, which is involved in providing information about changes in muscle length, and the Golgi tendon organ, which is involved in providing information about changes in muscle force. In the somatic senses, the sensory neuron is also the sensory receptor and responds directly to the stimulus. The axons of these sensory neurons may be myelinated or unmyelinated depending on the sensory neuron type. Next, let's look at the motor neurons a little bit more. Recall that alpha motor neurons of the somatic nervous system run from the spinal cord through peripheral nerves to innervate skeletal muscles in the periphery. Alpha motor neurons typically receive about 50,000 to 80,000 synaptic inputs, both excitatory inputs and inhibitory inputs. Alpha motor neurons are always typically active at some level. This produces a resting level of muscle activity known as muscle tone. Now, something that's important is that alpha motor neurons can only excite muscles. So, if you want a muscle to relax or have decreased tone, you need for the motor neuron to be less active, that is, producing fewer action potentials. And this would occur by increasing the relative amount of inhibitory inputs onto the motor neuron. So by having more inhibition, we're going to have fewer action potentials, so we will excite the muscle less, and therefore we will have decreased muscle tone. It's important to keep in mind that alpha motor neurons receive input from a lot of cells not shown in the simplified diagrams typically shown in your textbooks. For example, they receive input not only from sensory neurons, but also interneurons within the spinal cord called local circuit neurons, as well as interneurons descending from the higher brain centers, often called upper motor neurons. The third and last type of neuron is the interneuron. Interneurons are located entirely within the central nervous system. They conduct signals between other nerve cells. So, for example, they can receive information from either a sensory neuron or an interneuron, and their target can be either a motor neuron or another interneuron. Interneurons can also have either excitatory or inhibitory effects on their targets, unlike alpha motor neurons, which are always excitatory, and also sensory neurons, which are typically excitatory in most cases. So we could talk about excitatory interneurons, meaning the interneuron has an excitatory effect on its target, that is, it moves the memory potential of its target closer to the action potential threshold, or inhibitory interneurons, meaning the interneuron has an inhibitory effect on its target, that is, it moves the memory potential of its target away from threshold. That concludes the screencast on the three types of neurons. If you have any questions, please bring them to recitation or office hours.